Hi, everyone. This is lecture 4 of the Synthetic Biology High School Workshop Series. Today we're going to be talking about biophysics. It looks like a good amount of you managed to find the YouTube video that we posted for lecture 3 last week. Today, Juan is going to be teaching. Juan, you want to introduce yourself?
Hi, everyone. My name is Juan. I'm a sophomore at Columbia College. I'm currently studying biophysics and philosophy, so I'm very excited about this lecture, and I hope that you guys discover this field that it is, although it's not widely known, it's a very interesting field for a lot of people, and there are a lot of cool research going on in biophysics as well. So let's start by answering what is biophysics. So how I would define biophysics is just the analysis and the research on how life works through physical concepts, physics engineering, and physics modeling. What does that mean? That means that basically mathematicians, physicists, computer scientists, and chemists, all of them work together to understand life from a different perspective that just normal biologists do. And with that, we have a lot of fields that we can explore. So the first one is structural biology, which is basically the study of structures in the cell or different molecules through different physics techniques. So we use concepts such as Newtonian mechanics, electromagnetism, waves behavior, and even statistical mechanics and Brownian motion to design methodologies by which we can study certain structures in the cell. The next one is computer modeling. As I told you, biophysicists also use physics techniques and physics softwares to understand how molecules interact with each other. So for instance, they use a process called stochastic processes, where they analyze one molecule and then they see how this molecule reacts to certain treatment, and then by analyzing the same molecule, the several single molecules several times, they can make probabilistic models of how things behave in the cell, which is very similar to how Brownian motion works, for instance. The same with molecules in motion. And then we also have the more widely known part of biophysics, which is, first of all, neuroscience. As you know, neurons transmit electric impulses, and the study of these electric impulses can be seen as either direct current or can be also seen as capacitors. And in that same sense, there is also a lot of things to explore in bioengineering, nanotechnologies, and biomaterials on how to both study the brain and how to use the same mechanisms through which life works to apply to other things to create biomaterials. Another application is also medical applications. So for instance, radiation therapy, pacemakers, kidney dialysis, all of these have a lot of physical concepts that need to be analyzed before they can be used to treat patients with these conditions. And biophysics plays a key role in understanding how life works in this part. And the last part is also stuff related to environmental engineering. So for instance, there's been a greater push for use of more green friendly fuels in replacement of the traditional hydrocarbons. And one of these is for instance, biofuels. So a great area of research in biophysics is also how to harvest energy from living organisms to carry on the activities that we do every day. So for this lecture, I decided to focus especially in the first aspect of biophysics and the one that is mostly known, which is structural biology. Why? Because I think it's very relevant to what we're living today here with coronavirus, and I think it will illustrate a lot of physics, a lot of physical, chemical, and biological concepts through these phenomena that we're living here with coronavirus. So if you don't know coronavirus, the real name of this virus, or the one that we are facing today, is SARS coronavirus 2. And it's from the same family of the SARS coronavirus that also caused a pandemic in 2002. However, for that pandemic, we only had 9,000 people infected worldwide. And it was a much less traumatic event as the current pandemic is right now. These are numbers from Tuesday, April the 3rd, I think. And in Tuesday, we already had 1,400,000 people infected worldwide. So these statistics are terrifying, but at the same time they are very encouraging from a curiosity perspective. I think it generates the question: why, if these two viruses are so similar, have caused such different effects in how the world works? And this answer can be explained through biophysics and structural biology. So basically, how the coronavirus pathway works is we have this capsule of lipids that is surrounding genetic material that is RNA. And this capsule with lipids with RNA inside has some proteins that can communicate with the cells in our body. These proteins are called spike proteins, and this is why this virus is called coronavirus, because these spike proteins form a shape of like a halo, which in Latin translates to corona. And these spike proteins can communicate with a protein a receptor protein that we have in our lungs, also in our kidney and in the blood, but specifically in our lungs, because this is how we obtain the virus through respiration, which is called ACE2. Both coronavirus work both SARS coronavirus work through the same pathway. However, the SARS coronavirus 2 spike proteins have 10 to 20 times more affinity to these receptor proteins ACE2 in the lungs than the previous SARS coronavirus. So how do we know this? And the answer is through structural biology, specifically a technique called cryo-electron microscopy. But before getting to that technique, which allows us to see and study virus and vesicles that are more or less around 100 nanometers big. I want us guys to go before that. How do we study molecules, small molecules? How do we study proteins and protein complexes? And then we will arrive on how to study viruses and how we're currently have these technologies developed through the study of biophysics. Just for reference, 100 nanometers is a hundredth of what a normal eukaryotic cell is, which is around 10 micrometers. 100, no, 1,000 of what a eukaryotic cell is. So it's very, very, very small. And the normal light microscopy techniques are not enough to study viruses. So to start with, I want to start with how do we study small molecules? And for that, I want to give space to Miranda, who is going to talk about NMR spectroscopy.
Cool. So if you go to the next slide really quick, the first biophysical technique that we're going to talk about is NMR spectroscopy. So NMR stands for nuclear magnetic resonance. And the first thing that we're going to cover when we're talking about this technique is just its basic basis in physics. So protons in a nucleus have a spin magnetic dipole moment. So a spin magnetic dipole moment is a characteristic of a proton that is associated with the proton spin. So we represent this through the character mu, which is this little guy right here that looks like a U with the Z subscript. So when a proton is put in an external magnetic field, which in this case we will call B, the proton's magnetic moment can assume two possible states. If the proton is plus U, it's oriented in the same direction as the external field B. And if it's negative U, it's facing in the opposite direction of B. So there's an energy associated with the orientation of a magnetic dipole moment in an external field. And therefore, there's an energy associated with each of the states of mu. So when mu and B are oriented in the same direction, this is called the spin up state. And it has a low energy, which if you look at the right-hand figure, if you look at A, that would be the low energy or spin up state. If you look a little bit to the right of that, we can have, if U and B are in opposite directions, then it's called the spin down state, and there's a higher energy associated with it. the energy difference between these two states is equal to 2 multiplied by the magnetic dipole moment mu multiplied by the external field B. So you can see that in this first equation that we have right up at the top there, where we have delta E is equal to 2 mu B. This is also illustrated in the portion of the diagram on the right hand of the slide under C, where you can see the gap between the energy of the spin down and spin up states is equal to 2 mu B. So if we place a sample of something, say water, in a magnetic field, then the protons in the hydrogen portions of that water would tend to be in the lower energy state. But these protons can jump to the higher energy spin down state if they absorb a photon with the energy HF. So if you take a look at this equation underneath the first one, we have HF is equal to 2 mu B. So HF stands for the energy of a photon. If you can have the energy of a photon that is equal to the energy difference in between the spin down and spin up states, then that proton is able to absorb the energy of the photon and switch between each state. This is called spin flipping. And when we have a frequency, which is the F in this equation, we have a frequency of magnetic radiation that allows it to be equal to this energy difference in between the two states. We call this magnetic resonance. Generally, the photon energy that's required for magnetic resonance have a frequency in the radio wave range. So we can use NMR spectroscopy to generate a chart that represents the specific fingerprint of a given molecule. In this case, we can put a high concentration of a molecule into a solution and then subject it to varying frequencies of magnetic radiation and a varying strength of the external magnetic field B. So given a particular frequency of radiation, we can sweep through a range of different external magnetic fields that will show a resonance peak when B sweeps through the value at which spin flipping occurs. So this would be for a given frequency of radiation, if you sweep through different values of B, you would get a peak of energy absorbed when B reached the value at which this second equation, HF equals 2 mu B, is true for that particular proton group. So if you look at this chart on the left, we can see the NMR spectrum of ethanol, which consists of 3 groups of atoms. There's a CH3 group, a CH2 group, and an OH group. So protons in each group can undergo magnetic resonance. However, each group has its own magnetic resonance value of B. This difference in between their magnetic resonance values is why you have distinct peaks on this chart at different values of B exterior. So whenever there's a peak, which you can see is when the energy absorbed goes up, that is when you reach a magnetic resonance value. So magnetic resonance value is not only determined by the specific proton and by the external field, but it's also determined by the other things that are surrounding your nucleus of interest. So in this case, that would be the electrons that are around your nucleus, other nucleus groups or proton groups that are near the nucleus that you're studying. So because each nucleus is situated within its own magnetic field, we will call this field B internal, that are generated by the other nuclei close to the nuclei of interest, as well as the arrangement of electrons around the nucleus. The resonance peaks form a unique NMR signature by which ethanol can be identified due to its specific structure. You want to go to slide 2? All right. So there are a lot of uses for NMR technology, but we're most interested in its use in biology. So once we have the one use we have for NMR spectroscopy is for protein visualization. So it's super important for biologists to know the 3D structure of a protein because protein structure determines function. Active sites and binding sites on proteins are often very specific for what they bind to, and this specificity is determined by the protein's specific 3D conformation or its specific shape. So NMR can be used to reveal the atomic structure of macromolecules in a solution. That's why we think it's useful. So the type of NMR that we talked about on the last slide is a 1D spectrum. It's useful for determining the nuclei groups that are found within a single molecule. The next step in protein visualization is for us to attain a 2D NMR spectrum by something called nuclear Overhauser enhancement spectroscopy, or NOESY. So NOESY displays pairs of protons that are close in proximity, even if they aren't close together in the primary structure. So when we think about proteins, proteins aren't just a long chain of amino acids. Proteins are folded in very different specific ways. And for us to truly understand how they work and how they interact with other proteins and their environment, we need to know what they look like when they're folded, not just when they're a chain on a sheet. So when nuclei are close together, they have a magnetic effect on each other. This is called the nuclear Overhauser effect. So magnetization can be transferred from an excited nucleus to an unexcited one if they're less than 5 angstroms apart. 5 angstroms is an incredibly small space. It's less than really anything that you would be able to see with your eye or even under a microscope. The interaction between nuclei is proportional to the inverse 6th power of the distance between them. So basically, the smaller distance in between the nuclei, the larger the interaction. So the NOESY generates a 2D plot that's shown on this left side of the slide. We're going to look at this simplified one first. So we can see a diagram right here of what the folded protein looks like. And this is a very simplified protein, but we're going to use it just to demonstrate what the NOESY will do. So as you can see, we have 1, 2, 3, 4, 5 proton sites or nuclei sites on here that we're interested in. And you can also see that the way that this protein is folded, sites 2 and 5 are very close to each other in the folded structure, even though they aren't close to each other in the actual chain. So right next to that, before we get over to the right side, if we look just in the middle of the page, we have the NOESY spectrum for this protein on the left. So the way that you read this is a little bit more complicated than the first one. But how you do it is the diagonal line up the plot shows all of these nuclei in order. So we've got 1, 5, 3, 2, and 4 that are all going up in a line. So you would expect to see this on any NOESY plot, regardless of how close or how far each of these are to each other. Our baseline generation is going to be this diagonal line up the top. So as you can see on this diagram, there is something called off-diagonal peaks. So this would be these 2 pink dots that don't fit in with the diagonal line of the rest of them. So these off-diagonal peaks, if you go both up from the diagonal peak to the diagonal line, and if you go sideways back to the diagonal line, in both cases, you'll see that this pink dot, when traced back up to the side, down, whatever, to your diagonal line, it shows the intersection between where 2 and 5 are. So what this means is that 2 and 5, because they produce this off-diagonal peak, it indicates that they have a close relationship and that they are, in fact, near each other, as is shown and confirmed in this protein structure. So in general, we'll call this close relation less than 5 angstroms apart. And this diagram on the left is very simplified. So if we look at this one on the right, this is the NOESY spectrum for a 55 amino acid domain of a protein. So each off-diagonal peak, which would be all of these little dots that are represented in pink on this spectrum, each off-diagonal peak corresponds to a short proton-proton separation. So that would be where 2 nuclei are very, very close together with each other. So there are hundreds of these interactions on the spectrum, and these proximity relations can be used to reconstruct the 3D structure of a protein. You want to go to the next slide? All right. So how do we use all of that information that we found? So we can use this information about which nuclei are close to each other to be able to visualize protein folding better than we could before. So on the diagram that is shown, we have the points on the unfolded protein that are connected with these little pink lines represent areas found on the NOESY spectrum that are identified as close together. So these connection sites would be places where we had that off-diagonal peak. So when we have enough of these spatial constraints, it's possible to visualize the 3D structure of the protein. This usually isn't done by hand. Most of this is done by computer programs because, as we could see in that even 55 amino acid section of a single protein, there were hundreds of places where the nuclei had interactions with each other. So trying to do it by hand would be really tedious. So a computer is usually used for this portion of the visualization. But as you can see, on the top area where we have the unfolded protein, which has these interaction sites and these pink lines that are indicating which sites are interacting with each other. If you can use visualization technology, you can get something down to this calculated structure, which is where all of those sites that are identified as being close to each other are mapped out so that we can determine a specific protein structure that might be viable. So when we have enough of these spatial constraints and we can visualize the 3D structure of the protein, that still doesn't mean that we know the exact structure of the protein. So rather than definitively finding a single 3D structure, most people will seek to generate a family of structures, and that can be shown on this black and pink diagram on this side. So there are a few reasons that we don't want to decide on one single structure and say, like, that's it, that's what the protein looks like. So the first thing is that there are so many spatial constraints that we have. And we didn't even talk about all of the different types of ways that you can try and figure out the shape of the protein. This example simply talks about finding out which nuclei are near to each other. So there are a lot of other things that can affect the overall shape. There's constraints with bond angles. There's constraints with which things repel. There's like a lot of different things that we could talk about. So one of the reasons that we try to generate a family of proteins is that we don't have enough constraints experimentally accessible to fully specify the structure. So a second thing that's a little bit unpredictable with NMR is that the distances obtained from the analysis of the NOESY are only approximate. So we know in general that all of these interactions are going to be less than 5 angstroms apart, which is pretty specific. It's a very, very small thing. But all of the atoms that we're talking about are also incredibly small. So although there has been some advancement with specificity with regards to NOESY spectrum, where we can determine whether it's a strong, weak, or medium signal, which corresponds to a different distance in between each nuclei, we're still not able to show the exact specific distance in between them. We can really only guess. And because the protein folding is in general so detailed, even a little bit off can lead to a whole different structure. So since these distances are only approximate, that's another reason that we go for a family of possible protein structures instead of just one. So the last reason is that experimental observations when we're producing an NOSEY spectrum are made not on a single molecule, but on a large number of molecules in a solution. So they may all have slightly different structures at any given moment. So because of that, when we're doing visualization, we're not identifying one single protein and looking at every single nuclei within that. We're taking nuclei of several different areas of several different molecules of the same protein. And so since each one might be a little bit different, we're generating a family of possible structures instead of a whole one. So on this diagram on the right-hand side, if we look at all of these black lines represent each individual visualization. And if you follow the bright pink line around, that's what the average of all of these visualizations looks like. So that would be the one that we would consider the most likely representation of the family. All right.
Okay. Thank you, Miranda. So as you can see, NMR allows us to have a resolution of 5 angstroms, which is pretty small. Is 1/10 of a nanometer, and that's a great resolution to study molecules. But as Miranda said, molecules are floppy, and NMR has some trouble, especially with this floppiness that Miranda was describing at the end, because in a substance, there exist many ways in which a molecule can be, especially because bonds themselves between atoms rotate. And if these bonds rotate, then the whole shape of the molecule can be altered, and these data that we get from HNMR and in two-dimensional can get very floppy. So for that, scientists designed a very long time ago a way to study this type of molecules called X-ray crystallography. X-ray crystallography actually wasn't first used to study biological molecules. Um, why? Because X-ray crystallography was mainly used to study other types of chemical molecules, such as salts, um, um, other things, other metals that form crystals, and other type of non-relevant molecules. But then around 1905, when Watson, Crick, and Rosalind Franklin realized that they could use this technique, X-ray crystallography, to study DNA, and that's how they discovered that DNA was actually a double helix. But before going into all of this, I want to understand why X-rays? Why are they useful to study biological molecules, and what is the biophysical concept behind it? So X-rays are just another type of light. So light is basically exists in a duality, and this duality refers to light is once a wave, and it's also a quantized particle. What does it mean that it's a quantized particle? It means that it can only acquire certain energy values. But before going to this definition of the quantized particle, I want first to understand this behavior as wave. So as a wave is what we call an electromagnetic wave. That means that it vibrates in the electric field, and it also vibrates in the magnetic field. And as any wave, it has a peak, a throat, and a distance between the peak and the throat. This distance we call wavelength. And due to the wavelength is how we define the different types of lights that we have. So the light that we're most familiar with is the visible light. That has a wavelength between 350 nanometers to 800 nanometers. And this is the light that we interact with every day. This is the light that our eyes can see, and it covers all of the colors of the spectrum. Bigger than this type of light is what we have called as infrared, which is basically heat. So if you have ever felt that when you're close to some fire you feel warm, it's because fire itself is emitting this infrared electromagnetic radiation, and we perceive it as heat. Even bigger than that is, for instance, radio wave, and for instance, TV, FM, or AM, which are even bigger than a human. We see here wavelengths of 100 meters. But on the other side of the spectrum, we have things that are very, very small. What is the main characteristic of these waves, of these electromagnetic waves? Is that they are very energetic. Because they are very energetic, they are not usually used by scientists because they can be very harmful to biological materials. An example of this is ultraviolet, which is the type of light that we get from the sun, and that can cause sunburns, and that can also lead to denaturalization of skin cells and lead to skin cancer. But more dangerous than that is even X-rays. That's why when you go and take an X-ray imaging, usually doctors put LED in front of you of the parts that don't need to be visualized, because X-rays can be very harmful to you, especially to your vital organs. So we want to avoid the amount of radiation that your body acquires. But what about the fact that life behaves like that? Why is it important to us? And specifically, why is it important for us to study biological molecules? So for that, we first need to study wave-like behavior, which is diffraction. So diffraction is the phenomenon through which light passes through place where it cannot, where it is blocked. So as we see here, we have in the left, we have these wavefronts of light that are coming on this side, on this left side of the barrier. This is similar to when you have a light on your room, and therefore you see, and when you are, and then you go out of the door, and then you see that there is a shadow of light, and that light doesn't go around and occupy that part of the shadow. We see that light only diffracts through specific spaces. That is, that it only passes through a small slit, and then from that slit it expands. And this is an interesting behavior because we can think of these slits as also other type of matter. It doesn't have to be a thing that you can touch, a thing that you can see, but with these very small wavelengths, we can think of these just as matter, as atoms. Another important aspect of diffraction is that if you understand this from a very minuscule perspective, you can think of these as each of these spaces is a new source of light. So imagine this source of light expanding again after it passes through the slit. As it passes through the slit, it saves the wavelength to different points. And depending on where that wavelength arrives, we will see some phenomenon called superposition. What happens when we have superposition? Take, for instance, here on the left, this diagram that we have here. We have one wavelength of light, which is wave 1, that has a certain amplitude, and it is in a certain phase. Then we have wave 2, which has the same amplitude, and it's vibrating in phase with the same wave. When we combine both of these waves, when we add them up, we see that we have a resulting wave that has twice the amplitude, and it has the same period of vibration. What happens then when we have the same wave with the same amplitude, but they are out of phase? So when we have a peak in wave 1, we have a throw in wave 2, and vice versa. When we have a throw in wave 1, we have a peak in wave 2. Well, if you add both of them, what we will see is that we will have a wave that has no amplitude, a wave that basically light cancels itself out. And this is what we see when light crosses a slit. When after light crosses a slit, each of these new sources of light can direct it towards a point. And if there is another source of light that when it gets here, it adds up, and they cancel each other, then what we will see is that there is no light, and we see this interference pattern where at the middle, we see a higher intensity of light, and then we'll see that it decreases its intensity, then it increases again and decreases again. And this is all caused by superposition of these new sources of light. This is a phenomenon that you can even do in your own homes. Just get a flashlight, get a very narrow slit, and then you will see this pattern of diffraction. So how do we use this light to study atoms? And the first thing that we had to realize, and I guess that you guys have maybe seen on your physics or chemistry classes, is that atoms, especially, are quantized molecules. What does that mean? That electrons are not just orbiting around a central nucleus. They are more like clouds of probabilities where an electron can be. So say, for instance, here in an atom that only has one electron, we'll see that that one electron can be in all these positions. But then if you add another electron, then this one electron can be located in this orange positions or possibility of positions inside another bigger cloud of electrons that is located out. And each of these random possibilities where the electron can be located are determined by something that we call the Schrödinger equation. This Schrödinger equation has a certain energy associated with it. So what happens when we increase the energy of these electrons is that these electrons are going to have another type of Schrödinger equation, and they are going to be farther from the nucleus. The way in which we give energy to these electrons is through electromagnetic radiation. So when Miranda was saying before, we irradiate light, in this case X-rays, to electrons, and they will increase their energy level. But electrons always, and nature in general, always want to be in the lowest possible energy state. Why? Because being in the highest energy state is very costly. So when electrons are in these high energy states, they will want to go back down. The amount of energy that they release when they go back down is the same that they absorb. So say if they absorb certain amount of energy from an electromagnetic wave, they will release a photon of the same energy and in the same wavelength that they received. I'm going to jump this. So this is what we see here. We have an incident X-ray. The X-ray is absorbed by the molecule, and then that same X-ray is re-emitted by the molecule when the electrons go down to a lower energy state. Think about this in Newtonian mechanics. So a particle of mass m moves towards moves at a speed u in vacuum, has a kinetic energy of the mass times the square of the speed divided by 2. Then that particle collides with another particle of mass m, and that molecule starts moving at a speed v while the other comes to rest. What we have here is just an example of a perfect elastic collision. In this case, the second molecule, which was initially at rest, gains the kinetic energy of the mass of the molecule times the velocity that it acquires squared divided by 2. The same thing happens here. The X-ray, basically, which is a photon, and it's that characteristic of the duality of light. Photons are basically these particle behavior of light, crashes an electron, and the electron absorbs the same kinetic energy that the incident. So LDP proposed that basically matter can be understood both in its matter behavior as something that has a mass and has a velocity and has a position in the more Newtonian mechanic perspective, but it can also be understood in the quantum mechanical perspective as something that has a wave-like behavior. He proposed that the energy associated with any particle is basically proposed by by any wave is basically the Planck constant times the frequency at which that wave is oscillating. In the case of light, for instance, we have that velocity is defined by, but that frequency of oscillation is defined by the Planck constant times the speed of light divided by the wavelength of that light. So the amount of energy gained by an electron in an atom will be equivalent to the wavelength, or be inversely proportional to the wavelength that is scattered by that atom. And another important thing that will come back later when we talk about cryo electron microscopy, but I want to just say it now, is that for everything that has mass and has a momentum p, which that means mass times volume, just the Newtonian relativistic momentum, that momentum can tell us about its wave-like behavior. So that molecule has a wavelength that is inversely proportional to both the mass and the velocity of that matter. What do we study when we study crystals? When we think about crystals, it's just we basically petrify molecules. How do we petrify molecules? We basically dry them up, and through different techniques, we organize them in a very symmetrical way and in a specific pattern, so that we can study them. So here we have an ice lattice crystal of water. So we have hydrogen here, and oxygen here, HO2, and these molecules are aligned in the same way as we can see here. So it is not how we had in the NMR spectroscopy, where all the molecules were in different states and they were sloppy, and proteins had like different orientations because bonds can rotate. No, here molecules are aligned and they have a specific orientation. It happens to be that we can do the same not only with DNA, as Rosalind, Franklin, Watson, and Crick did, but we can also do this with proteins. So what we have here in our right are basically protein crystals. I know they are beautiful, but this is what scientists use to study proteins for almost the whole 20th century, and which a lot of scientists are still using today because it's such a useful technique. So how does this technique work? Well, as you can imagine already, we irradiate a lot of X-rays to these crystals, to these protein crystals that through a lot of work we have put together in this specific state. When we irradiate these X-rays, uh, each of the atoms in a protein will act as clouds of electrons. This cloud of electrons will work as the barrier that we described before. They will again absorb this energy of the X-rays and scatter it at a specific wavelength and at a specific energy. What happens? That all of this energy will that it is scattered by the molecules will probably encounter the same energy because energy is conserved by other molecules as well, and they will all neutralize each other. So all these scattered photons by the molecules can almost be ignored because they will probabilistically, the most likely thing to happen is that all of the energy is scattered by these electrons because it's the same in all of the molecules, will cancel each other. However, in the spaces between these clouds of electrons that we have in each of the atoms, X-rays will pass through. And when X-rays passes, when the X-rays passes through these spaces, we will see what happens when light passes through a barrier. We will see an scatter pattern. And in the same scatter pattern, the diffraction pattern, we will see places where, because of superposition, we'll have a higher intensity of light, a lower intensity of light. And we can record this as these diffraction patterns in these very big films, which is what we have here down. So what does these places where we have higher intensity of light and lower intensity of light tell us? That light has passed through, and we see we can think of this as this can give us a clue of how the barriers are distributed around the crystal, which means how are these clouds of electrons distributed around the crystal. So again, this is the same example for one protein molecule. When we irradiate light to the protein molecule, we will see this scatter pattern. But when we have this crystal, we will see that we will have places where we have more intensity. Then we can take these diffraction patterns that we have gotten here, and through a series of mathematical analysis called Fourier transformations that allow us to take the intensity at each of the points and understand it as a single wavelength, as a single wave. Then we can further separate into smaller waves. We can create an electron density atomic model. So this electron density model, atomic model, allows us to think, well, there is a higher amount of electrons here than maybe here, and say that this is, for instance, the side chain of a molecule. Then, because this has a higher electron density, we can think of a higher of an atom that has more electronegativity here. For instance, oxygen or nitrogen. And with these predictions that are done by computers, we can actually help predicting the structure of the molecule. What is the advantage of this? Remember that molecules are oriented, or proteins are oriented in a very specific way. They're crystallized. So they are not sloppy and rotating all the time, which can be very annoying to study. So these electron atomic models can give us much better predictions what is the structure of a protein. Yes, it has less resolution than NMR. X-rays has wavelengths of around 10 nanometers, which is the lowest resolution that we can get, while in NMR we have 0.5 nanometers. However, when we work together with NMR, we can understand how these molecules are formed. And when we get together this information, we have a great understanding of what the structure of a molecule is. Um, However, this is not enough sometimes. Sometimes you want to study molecules that do not crystallize correctly. Say, for instance, viruses. Viruses are just these like balls of oil, which are, again, very sloppy to study, and you cannot get them to organize in a very specific way. So this is where scientists were faced with a very big new challenge on how to study these molecules more effective, or how to study these um, biological entities more effectively. And here is where they came with the idea of cryo electron microscopy. So cryo-electron microscopy, cryo-EM, or cryogenic electron microscopy, it's actually a very recent technique. In 2017, these three guys, Jacques Dubochet, Joachim Frank, and Richard Henderson, got the Nobel Prize for it. But the interesting thing is that they didn't get the Nobel Prize in biology or in physics. They got the Nobel Prize in chemistry. Why? Well, cryogenic electron microscopy is indeed a physical technique used to study biological molecules, but the way in which these all binds together is through chemistry. So I specifically put this example here is because this can show you a way in which all areas of science can be integrated to solve a very important biological problem. Specifically, Joachim Frank is a professor at the medical center at Columbia, and he does research at Columbia. So I guess we're all very proud of him as well. So what is electron microscopy? So electron microscopy is basically an electron gun that basically bombards our sample with a lot of electrons. These electrons are directed through different mechanisms. We have here like a specific coil that generates a magnetic field, and electrons will be like grouped in the middle. And electrons will shoot across the sample that we have. This electron microscopy has been a very widely used technique, and it has allowed us to have these beautiful images. Here we have a reflection electron microscopy where electrons are shooted at a molecule, and then they reflect back, and we have this image of a fly, of the head of a fly, which is incredible. And we also have these textbooks images of a mitochondria, for instance, which can only be gotten with electron microscopy. So why is electron microscopy useful? Why does it work? I took this example from hyperphysics. I guess some of you have used it for high school, but I think it's very useful to understand this concept. So let's bring back our friend LDB, which it's equation that says that anything that has matter can also has a wave-like behavior. So the wavelength associated with a certain thing that has a mass m and a velocity v, which means that has a momentum p, a Newtonian momentum p, is basically the Planck constant divided by that momentum. So how does this relationship apply to all particles? So here we have a baseball. It is pitched at 40 meters per second, and it has a mass of 15 kilograms. So with the LDB equation, we can basically have that this baseball has a wavelength of 1.1 times 10 to the negative 34 meters. This is an incredibly small wavelength, so small that it is completely negligible. That is why we don't see baseballs exhibiting a wave-like behavior. It's because their wave associated with its wave-like behavior, it's so small that it doesn't have an impact in our daily life, where we don't interact with this wavelength. Um, for reference, the diameter of an atom is 10 to the negative 10, so it's way too small. However, for an electron, which is accelerated through 100 volts and has a velocity of 5.9 times 10 to the 6, which is very close to the speed of light, we have that it has a wavelength of 0.12 nanometers. That is almost an Armstrong. That is an incredibly good resolution. Why? Because it is not very small for it to be negligible, but it also has the perfect size to study molecules. It is much smaller than a wavelength, a normal electromagnetic wave that has, like for instance, with X-rays that we have 10 nanometers. So it allows a much better resolution, and we can use these electrons in the same way as we have as we use light in X-ray in X-ray crystallography. We just shoot these wavelengths of electrons to a molecule, and due to its interaction with matter, we can understand the biological entity that we're studying. So after understanding electron microscopy, we need to study cryogenic electron microscopy. So this is a technique that they develop, and which is basically you take a molecule and you freeze it to very low temperatures. When you freeze a molecule, you're basically doing the same as you do with when you're creating crystals. You're making that everything stays at one specific place. Things don't move, things are not sloppy anymore, and therefore you can study them. Then you take this very cold sample and you put it under an electron microscope. And you don't only take one picture, you actually take a movie of many pictures. And with this movie, and again with computational softwares, you can actually recreate the structure of a molecule. So what we have here is a low-density lipoprotein, which is a molecule that is used by cells to transport cholesterol across the body and to the cell. So what we did or what scientists did in 2017 was that they took this low-density lipoprotein, which is a lot of lipids associated with a molecule called a protein molecule called ApoB, which has a lot of cholesterol inside. They froze it. They took this movie. They predicted the structure, and now with that, we actually know much better how this low-density lipoprotein is. We know the different domains of ApoB. We know where these domains associate with the different lipids that constitute the LDL molecule, and we have a much better understanding of low-density lipoprotein. Why is it important? Because low-density lipoprotein is also associated with coronary heart disease and other proteins associated with high cholesterol levels in the blood. So by understanding better how low-density lipoprotein works, we can also design better drugs for these real medical problems that we have in humans. So this is what concludes kind of our trip that we started at the beginning with the structural biology. I present you the spike protein of the SARS coronavirus 2. This protein was actually studied with cryogenic electron microscopy, and this allows us to see that because of the orientation of this molecule and the specific substituent groups, it has much better affinity for the ACE2 molecule in our lungs, and this is why this virus is much more contagious than was the previous SARS coronavirus of 2002. because through an analysis of structural biology, the structure of these proteins, we have come to identify that it can actually be much better at connecting with molecules that are in our cells. So this is very important because what it means is that through the study of biophysics, and more specifically about biophysics techniques in structural biology, we can understand how to define things work, why are things like they are, and we can actually calculate the risk of diseases. Two, we can also define new ways in which we can counter this coronavirus. So for instance, now that we know how this coronavirus acts and we know the structure of it, we can design, for instance, an inhaler that will block the ACE2 channels in the lungs and maybe help for a treatment against coronavirus. But this is, of course, all under research, and it's research that cannot be done without biophysicists. So I know that we have spent all of this lecture talking about structural biology, but there is much more out there to study in biophysics as just structural biology. It's just like the main field that biophysicists study. So I want just to give you another 3 examples of how biophysics is actually applied to understand biological mechanisms, and try to motivate you to also push your own research in these new fields. So the first one is, of course, systems biology, and is the same thing as in structural biology. Once we know the structure of a molecule, once we know how other molecules fit in that molecule, we can actually create models in which we can create a system, and those and how each of those parts of the system interact with each other. So for instance, here we have a ribosome understood as a motor molecule, as a motor that you see in cars or in different devices. Because we know the structure of each of these molecules, we can actually have much better models of how these ribosomes work. And we can, again, help to cure diseases or help to treat diseases. Other aspects of systems biology and biophysics is, as I explained before, how, for instance, we use thermodynamic data or just Brownian mechanic data to understand how these molecules work through, for instance, stochastic processes. Another example is neurobiology, but I didn't want just to use a human as an example. So here I have a shark. Sharks have these very interesting organs called the ampulla of Lorenzini. And what these organs do is just they have a jelly-like feeling that can detect magnetic fields and electric fields down to 5 nanovolts. What does that mean? That it is through these organs moving through an electric field that activate neuronal pathways in the shark. And for instance, because when we move our muscles, and that is an electrical signal, sharks can detect these in the water. So they can detect moving animals just because they are moving their muscles, and that creates an electric signal through organs, even though they cannot see them. They can also detect the magnetic field of the Earth, which I think is very impressive. So these organs get excited when they move through a magnetic field, just as a coil generates energy when it moves through a magnetic field. And this is how they can orient themselves and actually migrate through very long distances. And this is also the same or kind of similar to what whales and dolphins use in their magnetoreception and magnetoorientation in the Earth. And this is how they can do these beat migrations around the Earth. And talking about magnetoreception, there is a a growing interest of biology trying to be understood from a quantum mechanical perspective. This is still under research and is still very new, but there are many phenomena that, although they occur in a quantum level, they can or they may have some impact in how molecules work and therefore in how biological systems work. One of these examples, and which is still under study, is the cytochrome. Cytochrome in birds. Cytochrome is a molecule that we all have in, or most animals have in our eyes. And it is the one that is in charge of analyzing or blue light or incoming blue light into the eye. This cytochrome gets excited and releases a free radical. However, this free radical is associated, or it is thought to be associated through quantum tunneling through its electron pair. And this is how, when this electron pair is associated to quantum tunneling and it is put in a magnetic field of the Earth, how it can help proteins orient themselves within this, the eyes. And this is maybe thought how some birds have this sense of magnetoreception and they can also do these big migrations. So I think this was all that I have prepared for today. And I hope especially this excites you of how big and how beautiful can biophysics be, and especially how important biophysics research is nowadays, and not only for structural biology, but for other many important fields in biological research.
All right. So it looks like that's all for today. Thank you to everybody who watched this. As always, please feel free to contact the systems biology email with any questions or concerns. I know we talked about things that were a little bit different than what we normally do. We don't normally touch on anything physical or structural. So if you have questions, please feel free to reach out and we'll get back to you on those as soon as possible. Next week, we'll be starting Foundations of Project Design. Yeah, that's it. Cool.
Okay.
I'm going to stop the recording.